Do you have a
Yes.
sort of an idea which ones you got?
Yes. So one of them, before I forget, is the one on the eye.
Mm-hmm.
There's like a mass, a growth next to the eyeball. It
Yes.
says number 23. Is that like what we're supposed to know? I was trying to figure that out. What that is? Is it a muscle or
This puppy,
that
right?
one? Yes.
That's the lacrimal gland.
Oh, okay.
That's the lacrimal gland. So when you see that, you always know that's on the outside. So that's lateral on the eyeball. So you always know that's close to the temple on the outside. And that's where the tears are made. And then the tears go across the eyeball to the medial side. And then that's where they drip into the nose, into the nasal cavity.
Got it.
Through the
Okay,
lacrimal. So
that
you have
one.
the lacrimal punctum kind of language on the medial side, and the lacrimal gland is on the lateral side.
Lacrimal gland. Okay, that one.
Perfect. Very good question. And you got the muscles figured out?
Yes.
Okay, good. Good. And if anybody else has something to add on or a question to these models, speak up. We're going to do a little bit of that while we start the class. Then I take roll after that. Oh my God. Yeah. Okay. Any other one, Anna, on the eyeball?
Yes. So on the ear.
On the ear. Okay.
Yes.
Oh, that's all.
That would be.
Yeah, the ear is a little goofy here.
Like the incus and the malleus. Where is that?
Mm-hmm. And the stapes, right?
Yes, yes.
So you got the incus is attached to the eardrum, right? And so you can see that. You can kind of make up that. No, the malleus is attached to the eardrum. I made that mistake.
The eardrum and.
Yeah. The malleus is attached to the eardrum. That's the first on the list. So that's when, if I point to one like this one, that would be the malleus.
That's the malleus. Okay.
All right. This, and you see how it's, I don't really actually know what this piece is, but that's attached to the eardrum. And then to the malleus, the incus is attached to it right here, the long piece here.
Okay.
And then the stapes looks like, the where you put your foot in and do horseback riding, kind of to the side. That's attached to the oval window here, which is then part of the inner ear.
Got it. Okay.
So you could see this piece is the stapes, and then this middle section is the incus, and then the first thing attached to the tympanic membrane is the malleus. Mm-hmm.
Okay.
Yeah, and if they are too hard on the test, too ambiguous, I've tested them many times, these questions, but it always can show up. Then you just communicate with me, and if it's too ambiguous, you usually win the point. So I'm not going to do. I don't want you guys to be too concerned or too anxious about making up what structure is. I try to be as clear as I can be, and if not, please communicate with me, and we'll be fine. Good. What else on here? You got some other ones on here, Anna?
No, I think that was it. I think I was looking at the picture totally different or wrong. I'm not sure why I couldn't figure it out. Now I see it.
No, I actually just had a class at Merced, and we had the same discussion. It's not clear. It's, this is the whole ear so you see the external canal, you see the tympanic membrane, okay, here, you cannot see the malleus, and then it gets fuzzy back here. And then we look it from the top, and that's where we pick it back up here. And so that's the cochlea, the semicircular canals, and I think that's it for the list.
Yeah, okay. Yeah, now I see it.
Excellent. No, that's good. Okay. Anybody else have some questions to these pictures since we're talking about them? Or actually, let me point out how to get them in case you haven't found them, because I put them up, I don't remember, last week at some point. So if we go to the class, where am I here? Dashboard. You can see my internet, right? My canvas.
Yes.
Yeah,
Yeah.
I
Okay.
can
Good.
see it.
Yes,
Good.
yes.
So when you go to the canvas here, modules, test 3. Maybe there we go. All the way at the bottom by test 3. Review. We get to pictures used on test 3. That's where I put them behind, and then you get a PDF out of it. And so those are the pictures. Again, it might not be as great because it was sort of an afterthought of doing it, but those are the pictures that I use on the test. Okay, so that's how you can access them. And so at the end today, we'll have us a quiz with those pictures, but for you at home for the final exam, what I will do is I will print them out if you can, and then label all the terms that you can find on those pictures. Some of them you can't see, but you know, for the majority you can get that. So that's how I will prepare, and you can, you know, on the test review, there's also those videos. You just watch those videos, and they will get you, they'll get you the answers. And I think most of the terms on the list are on the videos. Maybe a few of them are a little bit off on one another, but that's, you know, for the most part, they should be matching which terms on the list and which terms discussed. But so today we're gonna first go through the test review sheet. That's the 4-pager. That is the written part. And with that, we go to PowerPoint here. And you could see my PowerPoint. Is that, can you?
Yes.
Yes.
Yes. Yeah.
Good. So the first part is the sensation, the chapter on sensory receptors and such. And when I read there, receptor classification is done by stimulus type and or by structural complexity. And so that's how we can start. So basically, with this chapter, we try to talk about how can we translate the outside or the inside world and make them alive for us. So how can we experience that? And that is through the nervous system, a lot of it, because when we touch or when we feel warm or cold, that all gets picked up by the nervous system, and then the brain interprets the thing. What does it mean to us, and makes a picture of it. And so that sensation is the awareness of something happening, and the perception is the interpretation of that. And so that's kind of important to differentiate those. And then we go into what different receptors do we have. And so there we can then classify them by what type do they code. Are they for warm, for mechanical, like pressure and touch? Are they for chemicals or for light, the photoreceptors? Or are they pain receptors, which will be actually there's the type. Pain receptors is the nociceptors, and a nociceptor, noci refers to damage. So tissue damage creates the pain sensation. So that's kind of, I think that's actually an important point to know, that because, you know, that when you, that speaks against just popping pills when you have chronic pain, for example. There's something is raunchy somewhere, so we got to look a little deeper than that. So noci is pain, and then the structural complexity is either they're either simple or they're complex. And the simple ones are sort of here. They are here. They're like free nerve endings, like the pain, heat, and cold are coded that way. When they're exposed, then they send a signal. We have light touch in there. That's also fairly free. But then we get these onion-looking shaped things for deeper touch or pressure, where when these layers of cell membrane connect and touch, then we send out a signal for like, look, that was a deep pressure, not just a superficial touch. And then, so we have those that are fairly simple in their construction, and then we have the more complicated ones. And that's then the hearing, the vision, the smell, taste, and the stuff, the special senses. When we look at the simple ones, you know, fundamentally we can have them be either free, unencapsulated, or encapsulated nerve ending. So that's kind of like your fundamental differentiation between the simple ones. So that's between the pain fiber is a free, or a pressure is a encapsulated nerve ending. Good. So that's for that. And then the next point there has, well, then we have a slide on adaptation. And you know that, and you take a shower, you know, the first, it's like the water, depending on who takes a shower, but it doesn't change. It changes during the shower. It often gets warmer and warmer because we get used to the temperature. And so that's adaptation. And then some nerve fibers adapt, some don't. Like, for example, we have warmth and temperature that can adapt. We have some pressures, like if you put on a sock, you feel the sock on your foot, but after a while, your system just integrates it, and it's not a constant stimulus. There you got a sock on your foot. Yes.
Does the adaptation change depending on the age?
You know, that's a very good question. I am not sure what part changes as we age. Is it that we have less, less sensation, or, less nerve, or is it the adaptation itself? I don't know that. That's a very good question. I would think the adaptation in itself should not change too much, because we need to be able to get out of danger. Like, we need to be able to feel like, if it's, that we don't burn ourselves if it's, scalding hot or something. We need to be able to do that. But I can, generally I would think you're right, because everything slows down a little bit as we age. I'm just not sure if it's the nerve adapting, or if there is less nerve less nerve energy coming in. They're not firing as strong, stuff like that. And then some are not adaptive at all. Like, for example, if you walk and you move, your body needs to know where you are in space, and that's proprioception. And that, for example, is not adaptive, because you always have to monitor where you are in space. That those cells should not get used to a stimulus. That's kind of the adaptation. We're getting used to a stimulus, right? And then the other one that's not adaptive either is the nociceptor, the one that is tissue damage. You're not gonna, hit your thumb with a hammer, and then the second time the thumb goes like, oh, well, we felt that before, we're used to it now. And then you hit it again. That would be a bad voodoo. And there is conditions where you have that. And there's, like, for example, I forgot what it's called now, but there's a condition of people, then they walk with a twisted ankle, and they keep walking, and they don't notice. And within no time, that joint is destroyed.
Right.
So that can be very dangerous. So that's adaptation. And then the next thing that we talk about is receptive field. And that's like how many receptor cells are responsible to monitor a specific surface area. And so you can plot it out, and you can say, look, when we actually measure all the fields, and you can measure that with a 2-point discrimination thing. So you can take, a paper clip or something that measures it a little more scientifically, and you can poke a person in 2 different points. And if they can identify them as 2 different points, you know that you have 2 different nerves firing from these 2 different areas. If you have them closer and closer together, there is going to be a point where the examined person, the patient, will only feel 1 point, even though you have 2 points. And then you are only having 1 nerve that fires off from those 2 places into the brain. And then you can sort of plot out the receptive field if you want to do that around the body. And so a large receptive field gives us fewer nerves that go to a given area. And so that gives us a low resolution impression in the brain. So, for example, in the back, you don't need to have a real super sense of fine-tuned touch in the back like you need in the thumb, for example, where you need to feel, where you need to have an ability to put your hand in a pocket and feel which coin is this, where you really need to be organized. The back is not as important. It's important, but not to that degree. And so that's where we can then go back to the sensory, that was in the brain, the post-central gyrus, where we map out all the areas of the body, which ones are represented in that primary sensory cortex. And that sort of goes together also with how many nerves go to those areas. So you will experience that on that gyrus, the post-central gyrus, the area for lips or for a thumb is really large as compared to the area for a back in terms of representation of those nerves. So that's what I wanted you to know about the receptive field. And then from there, we go into the eye. And we talk about different structures. We have some of these are anatomy terms, where they have the eyeball and the cornea and the sclera, and then the choroid, and then the retina. So we have these layers, and we talk about them, I think, somewhat in the lab. Yeah, not the different layers. That's more in the quiz. So the eyeball is round shaped, it's a globe, and it has a tough outside layer, and where we see white, that's known as the sclera. Where we see clear, that's the cornea. And then we got that's the fibrous tunic, the outside layer. Anything on the outside is usually fibrous, because it's got to give it shape and all that stuff. And then the middle layer here is the choroid and the iris and the ciliary body. And so that, when you see the color of the eye, that's the iris. So that's part of the vascular tunic. And then the choroid is what brings the, it takes some of the light reflection on the inside of the eyeball up, but it also gives us blood supply to the retina and the eyeball. And then the retina is the yellow layer for us. Nervy is yellowish, not that it looks yellow. But
I have a question.
Yes.
On the previous slide, one of the diagrams has an arrow, like 2 arrows pointing to the sclera, but they're to different parts of the eye. And I was confused when I was labeling if like this is incorrect or if it's just in 2 places.
Look at that. Look at that. Huh. Got it. And so that's supposed to be going to the whites, but I will put that arrow further back, because that, as soon as it's rounded, I kind of, well, it probably about here. So that's the outside layer. This one, I would grab this out. I'm not totally sure what it's trying to go to, because that's more looking at the chamber. So take this out. I got to make a note of that. Thank you. God, I tell you, the stuff, doing these things, there's so many little tweaky mistakes and things that I can never get all of them out. So I would not worry about this part here. I would go right to here, but put the arrow a little bit more where it's round of the eyeball. Thank you for that.
Thank you.
And see, this is talking, see here, it says vitreous body. And so it's talking about the vitreous body, the vitreous humor. So the front, I think what they meant to label here might have been the anterior chamber. Right here, because they see this darker purple, that's the vitreous body. And so that's the back gel-like structure of the eyeball that keeps the firmity. And then in the front, where it's more this color, a little lighter, we have the aqueous humor in there, which is the liquid that gets exchanged. Yeah, so that's a little bit more that we're going to talk about. But I have a feeling since they indicated vitreous body, this is a mistake and should be saying aqueous humor in there, for example. But we have a slide here where you see that difference, where the back behind the lens, that area, that space is filled with a gel-like structure that kind of keeps the shape of the eye, but light can still go through. And the front part in front of the lens, we have fluid that circulates. that sort of helps nourish the eye. And that's the aqueous humor, and it provides nutrients and maintains intraocular pressure. So that becomes important actually if we have glaucoma, because one of the things that can happen is as this fluid gets created and then has to drain out of the anterior chamber, these canals can clog up. The canal of Schlemm, they're called over here. They can clog up, and that they can then increase the intraocular pressure. And so I think on that previous slide, that's what we wanted to indicate. So let me see. The retina is the pigmented layer and the neural layer, and the neural layer is what picks up the light, and then it translates the light into electrical impulses that then go through a few layers of nerve cells to reorganize themselves and make so the first decisions what we're going to do with that thing, and then it becomes the optic nerve and goes into the brain. So here we go. The rods and the cones are embedded in the pigmented epithelium at the back of the eye wall. Within them, light absorbing retinal molecules. So rods and cones are here. These are these color things. So when we go further down, where was that rods and cone thing? Right here. So the rods are sensitive to light, and they're sensitive to darkness. And so those are the ones that activate when it's darker outside or in our environment. And they can pick up light without it being too bright. The cones are responsible for color vision or day vision. So when it's bright, what happens is the rods, let me show you now what's happening in there. When light hits these rods or these cone cells, we have a retinal molecule detaches from the opsin, from the rhodopsin molecule. So we literally have a detachment of a molecule that's a light absorbing retinal molecule from the protein structure, and that sends up the signal that we need to fire an impulse that light hit that receptor. And so that's very, in some ways, it's chemical, but it's chemically, mechanically kind of a thing. And so what happens when darker light hits these cells, the cones don't fire. They are not reactive to this darker light, but the rods will fire off, and you can sort of see dark shadows or a little green. Actually, if you look for a color, that's kind of the greenish hue we can get in the darkness, or less light. And then when you go to a light room from the darkness, what's interesting is you then have to sort of use up all the rhodopsin in the rods and detach all the retinal from the opsin from them, and white them out, bleach them out, they call that. Because it's way too bright for them. And then at that point, the cones are the ones that pick up the light again. They're going to get, when the bright light hits them, they can send the signal out. But this is when you realize, when you get from a dark room to a bright light, it hurts. And you have to kind of close the eyes or cover them for a moment until you're used to. And that getting used to is when the rods are bleached out and all those retinal molecules detach. And then when you go from a light to a darker room, it takes time to be able to adapt to that darkness. And that's when all the rhodopsin is being regenerated in the rods, and by the, and it gets more accumulation. And at some point we have enough, and then we can sort of see better and better in the darkness. So that's kind of the cool part that I think there is, that it's a chemical detachment that makes us see, that sort of like sends the signal out that something happened. All right, so that's in terms of the eye. Let's see what else we have in the eye. The muscles we do in the lab portion. I think that's pretty good. You guys have some other questions for the eye there that we should talk about? If not, we're gonna go to the ear and talk about that for a little bit. When we look at the ear, we describe the organ of hearing and also the organ of equilibrium. And so we have to be aware of that. It's the hearing is in this area where we hear it, and the balance, the equilibrium, is in here. It's in the vestibule and in the circular ducts. And so you got balancing in all the three axes in the semicircular ducts, and you got acceleration, deceleration down in here in the saccule theatrical. Where is the saccule theatrical label? Oh, that's okay. It's down in here. It's those two. I just always forget which one is which. Anywho, what's important, I think, what's really cool about both hearing and equilibrium, as we talked in the eyeball is a rhodopsin, and here we talk about hair cells. And so if you actually take that snail-like, hold on, let's back up. In terms of the hearing, this snail-like cochlea is where we do the hearing. And what we do is to explain it, we roll it out kind of thing. And so when we roll it out, we have a few different chambers in there. We have an external outside chamber here, and these chambers are known as, they call them scala. And so the one on top is known as the scala vestibuli. That's attached to the oval window, and that's attached to the bones, the ossicles, and they are attached to the eardrum. And so the eardrum picks up sound waves coming from the outside, translating into bones, and then the oval window gets pushed depending on what's going on in terms of the sound wave. And so if it's a really low pitch sound versus a really high pitch sound, they're gonna be activating different parts of this this channel structure here. And so what happens is the waves are different heights, and this upper chamber is differently high, has different heights as it goes down or you know, around the cochlea. As it winds itself up, it gets smaller, and at the base it's larger. And so that's the top one. And then the bottom structure, the middle structure, is where we actually have the organ of corti, the organ of hearing in it. And the way that looks, if you cross section, it looks like that in there. And when you take a bigger view of that, it looks like this. And so you have a basic membrane, and then you have on top is a gel-like structure. They call it the tectorial membrane. And from the basilar membrane up, we have hair cells that stick up from those hearing cells. And they touch the membrane, the tectorial membrane. And so now let's say we have a sound that has a certain pitch to it, and it hits the top wall. And as it hits the top wall, this is bone, that's a hard surface. It's gonna go past that channel into the middle channel and vibrates that tectorial membrane and basilar membrane and bends those hair cells, and then it exits through the bottom channel, and there it just exits into space through what we call the round window. And that shaking of the membranes and bends those hairs, and when those hairs bend, then we send a nerve impulse that, you know, we just heard a sound. And the sound we heard, depending on where it crosses over to the middle chamber, gives us a certain pitch. So does that make sense? A little bit?
Yes.
It makes
Yes.
sense.
So I think that's really cool. So hearing and also equilibrium happens when we bend hair cells. And the seeing happens when we detach these rhodopsins from these rhodopsins from one another. So I think that's pretty neat. So let's see what we need more from here. No, I think that's pretty good in terms of the hearing apparatus. And I'm not going to have much on taste or anything on taste and smell, but that basically gets us chemicals in liquid, also is picked up by some hair cells that could then trap those odors and get trapped. But they have to be in some moisture that we can pick them up. But that's not going to be too much on the test. So from there, we can then pick us up and go to the respiratory system, unless you have questions coming up. And the respiratory system, if you look at the whole functioning, it's concerning itself with picking up, as a big picture, picking up oxygen from the atmosphere and getting rid of gaseous waste, such as carbon dioxide or other ones too that, risk gases. I also like the fact how the respiratory system helps maintain the body's acid and base balance, and it does that with the kidneys. It's very, very important that our blood pH is at a certain narrow range, and we have multiple different mechanisms in the body that can make sure that's the case. And so we have the buffering system that we talked about in the chemistry chapter. We talked about the acids, the bicarbonate ions that pick up hydrogen ions. And then in the lungs, we can get rid of the carbon dioxide by breathing it out, whatever, and sort of circulating the bicarbonate ions that way. And then the kidneys are here to do a slow, long-term balance in making the bicarbonate ions to help with the picking up acidity in the system. So I think that's kind of cool how those systems work together. And in the lungs, what we need to remember is just we breathe out gaseous stuff, and carbon dioxide is a gas. So that's good for that. And then when we look at the overarching functioning or structures, we have two big parts to the respiratory system. We have an area that delivers the air to the lungs, and then we have the area inside the lungs right here, the alveoli, that the gas goes from the air, from the atmospheric air, to the blood system and to the capillaries there. So the conducting zone or the conducting passages is everything down to the alveoli. And then what they do, they ventilate the lungs. And when we want to further take that conducting zone apart, we have an upper part, which is down to the voice box, to the larynx. And then we have a lower part, which is the trachea, until we get to the alveoli. And the alveoli is the respiratory zone, which is where we have that exchange. So we call that process then where the oxygen goes into the blood, we call that respiration, the movement of the oxygen. We call the oxygen going into the blood in the alveoli external respiration, and we call the oxygen moving from the blood cells into the tissues, we will be calling that internal respiration. So that's those two terms. So external is still inside the alveoli. It's not outside the body, but it's where the air goes from the outside to the blood.
Well, then from that picture that you have there, the alveoli is covered in capillaries that then travel to the heart, like pick up the oxygen and take it to the heart.
Yeah, you see also in this slide, you got a fine network of capillaries around them. So you really want to make it easy for the oxygen to diffuse. And it's a diffusion process. It's simply based on concentration levels. And if
from looking at this, and that's a really good image.
The
we
other
have
one?
both red and blue, which means there's an exchange of both oxygen and then carbon dioxide, right? Picking up,
Yes.
bringing in the
Yes.
oxygen and releasing that CO2.
Yes, and you see that right here on this slide, where on the deoxygenated side, we have, this is the partial concentration of oxygen and partial concentration of carbon dioxide. We have, look at the difference. We have a 40 millimeters of mercury. That's a pressure thing, right? And at the beginning and at the end of the capillary, when we have all the exchange happening, we have 104. So that's a big pickup of oxygen. The drop up of carbon dioxide is kind of interesting. It's much less. But if it's much more toxic if the carbon dioxide gets too high. And the body measures oxygen content by measuring carbon dioxide content in the system, which is kind of interesting. But that's where you can see that diffusion. That's where you can see from the blue to the red how it really gets bright red pretty fast in there. So that's pretty neat, right? And it all is just diffusion. It just goes right through it. So when we go back to the airways, then the upper airways are here to warm, humidify, and purify the air. So, and when we get down into the alveoli, the air is kind of like, you know, tempered the way we need to have it in there. You just go to a cold climate and you breathe in the winter with your mouth. You can feel how cold the cold air feels in the throat. I'm sure you have noticed that, some of you guys. Most passageways also are having a lot of mucus secreting glands in them and cells. And so that's the mucous membrane. There are goblet cells in there, and they secrete mucus. And the other thing and so that, you know, makes that humidifies it, but it also, what the mucus does, it traps foreign materials in itself, and so we can get rid of it that way, and they don't go into the lungs. So that's the cleaning process too. And the other thing I always like is the cilia that are sticking up on those cells in the trachea, and they move, and by moving, they move the material past the cells to the outside of away from the lungs, basically. And a lot of that, then, you know, we swallow and is in the gut, but because the stomach can just take care of it. And so that's pretty cool there. Let's see. Then the next thing we talk about briefly is the larynx there. So as we move down the throat, past the glottis, we get to the larynx, the voice box, and that's about here mid-neck underneath the hyoid bone, which is right here. And you can feel that. And the guys, you can see the Adam's apple in the front. That's the thyroid cartilage piece. We all have that. You just have to potentially push a little deeper. And in the front, you can see how that thyroid cartilage is nice and big, covering the whole front. In the back, it's sort of open. But then we have a cartilage underneath it, and that's skinny in the front, but then it gets nice and big in the back over here. And that's the cricoid cartilage. And on top of the cricoid cartilage, we got these erythenoid cartilage pieces, and they are attached in. They can rotate around their own axis. Actually, they get rotated by muscles around their own axis. Because what we have there, we have vocal cords that are attached. Do we have a view from top? We have vocal cords that are attached on those processes here, and then they go forward to the back end of the thyroid cartilage. And so they are basically here, going up and down in this space, in here in this space. And this space goes to the lungs, to the trachea actually afterwards. And what happens is, as we exhale, these vocal cords, they're like guitar strings, and they start vibrating. And depends on how taut they are, that gives us a different pitch, level of pitch. And so that's kind of cool. That's how that works. And so I wanted you to get a brief understanding of that process. And the space that is made by these vocal folds, when it's open, that's the glottis. And the glottis is where we then go down into the trachea And from there we get to the bronchi, and then we go into the lungs. Good, and that brings me to the serous membranes. If we don't have questions, that's the last topic there. And we talked about this briefly in the heart, or the chapter talked about it in the heart. We also have them around the lungs, and we also have them around the gut. In the heart, a lot of what that membrane does, it helps decrease friction. But in the lungs, it also glues the lungs to the chest wall. And so this is how that works. So remember, or maybe remember, the serous membranes are a continuous membrane system that goes around right around the organ wall, actually attaches onto the organ wall, and then it goes onto the chest wall, and here it's attached to the chest wall. And so you can visualize that when you take a fist and put it in a balloon. You see you have a membrane around the fist, a little balloon part around the fist, and then some space, and then it goes around on the outside. And in between that space, which is a potential space, we have a secreted liquid. We have serous fluid in between that space. And so from a naming perspective, in the lungs, it's known as the pleura, and the layer that is attached onto the lung is known as the visceral pleura. The other one that attaches to the chest wall, that's the parietal pleura. So visceral always refers to attaching to the organ, the viscera, that's the organ, and parietal is sort of the outside, and that goes to the organ wall, or the chest wall on the outside, not the organ wall. Good, and the cool thing about the lungs is that that space in between what's in there, that serous fluid layer suctions the wall onto the chest, the lungs onto the chest wall. And so technically speaking, they call that the intrapleural pressure is below the atmospheric pressure, and that creates a suction. And so now when we move the chest wall, the lungs will, well, the volume where the lungs sit increases. And since the lungs are attached to the chest wall, they suction air into the lungs because they get inflated, they get bigger. And that creates a decreased pressure in here, and that brings air in from the atmosphere. So that's inspiration right there. So I always think that's kind of cool how that happens. And of course, then the other side is, you get something in between the lung and the chest wall, or you get like air, if you worst case rib breaks or something, and you get what we call a pneumothorax, and you have to they have to then put a tube in and suction the lungs back to the chest wall, take that fluid or that air that got out in between. So that could be a bit of a problem if it happens, but that's, I think, why the serous membranes are so cool. And then lastly, but not least, for this part, we don't really have, I don't test you on this, but I want you to understand it. So it's not on the review sheet. But when you regularly sit there and breathe, you call that the tidal volume. And then if you take a deep breath in, as deep as you can, whatever comes after the inhalation is known as the inspiratory reserve volume. When you take a deep, when you exhale and then you exhale as fully as you can, we call that the expiratory reserve volume. And so when you then, from the fully expired, fully inspire, you get the whole vital capacity. That's how much your lung can take as volume. You get that as a measurement. And then you also have a residual volume, which is when we forcefully expire, some gases remain in the lungs that keep the airway open. And we don't, that's not an air that's exchanged. When you look at the vital capacity, you're looking at the most air that you can take in a full deep breath that is exchanged, that is useful in the alveoli, basically, or useful for this, functionally useful for the system that way. All right, so I think that should be good for here, unless you have any comments or questions. And oh yeah, look at that. I didn't even take you guys' roll. We should do that right now. give everybody some goodies since they're here. So let me do that right now, and then you guys can think of some more questions, and if not, we do a quiz. So I got Nairobi.
Here.
I got Sarah. I got Anna. I got
Here.
Scarlett. Yeah, just give me a quick here. I got Tenaya. I got
Here.
Cora. Emmanuel.
Yeah.
Adam.
Here.
Kyla May.
Here.
Kawhi.
Here.
Brianna.
Here.
Vanery.
Here.
Verenice.
Here.
Edviges.
Here.
Abril.
Hello.
Hello. I got Nelia. Jamuna.
Here. Jamuna here.
Caitlin.
Here.
I got Heymanot.
Here.
And please, if I ever misspell your name, I say in the beginning, please correct me. I
You're
got
right.
Alexis. Yeah, the good part is usually people butcher my name, so we kind of have something in common. One time I had a student and the first name was no name. It took me 3 times to realize that they didn't have a first name. I thought it was a real name. Anyway, Alexis, Benita,
Here.
and Narcy.
Just present.
Good.
Here.
All right, super. So now what I want you to do is take out a sheet of paper and we're going to do us a little practice quiz, like 10, 15 questions. Now let's keep it at 10. I'm sure you guys want to go home. Oh, you are home. Off the computer, I was going to say. All right, so we're going to go through the lungs. Oh, before we do that, let me show you the points in the lungs that we have. We don't have that much in the lungs. We got a thyroid cartilage. So that's right here, thyroid cartilage. We got a cricoid cartilage. That's below here, the cricoid cartilage. We got the epiglottis. Oh, look at that. You can't even see the darn epiglottis. That would be a flap sticking out. You got, I need to make sure there's not another module on the tests. Model to do. Hold on. Sorry about that. I'm just remember there might be an extra model. I'll take care of that till next week if it is. A model larynx. Okay, but we can see pretty much everything here except for the glottis, except for the epiglottis. And then we got the trachea here, and then we get into the bronchi here, or one is a bronchus. And then the alveoli, you can't really see. These are these little air sacs in here. So guess what? I usually don't mark them, but I want you to know them. Then we got the lobes of the lungs. We got the right lung has 3 lobes. It's got a superior, a middle, and an inferior. The left lung has 2 lobes, superior and inferior. If we have fissures, we get a horizontal on the right and then an oblique each on the right and the left. The right has 3 lobes, the left 2, because we got the heart that takes up some space. And then the area where the vessels and the bronchi go in and out of the lungs is known as the Hylum. And this here that looks like a skeletal muscle is actually a skeletal muscle. It's the diaphragm. And so that's all for that list. Good. So let's do a little quiz and finish up with that. So give me, please, on the heart, number 1, I would like you to tell me or write down what is this, when you look at the aortic arch, what is this second vessel there that comes out of the aortic arch? That's number 1. And number 2, and if I go too fast, you just stop me, okay? Number 2, I'd like for you to tell me this vessel here, this is an artery, this red artery here that feeds the heart and goes around where the ventricles are here in the front, goes around it. So that's number 2. Then number 3, Give me this blood cell here with the marble background, number 3. Number 4, we go into the brain. We have a half brain here. I'd like you to tell me this hind brain here, the cauliflower brain in the back there. That's number 4. And number 5, I'd like you to tell me this white stuff here, these connections between right and left hemisphere. That's connections between right and left hemisphere. What's this structure called? Number 6. Above this structure here, we have these little dots. And these little dots are nuclei, which are clusters of cell body inside the brain. Nuclei that code for reflex stimuli. One is for visual, one's for auditory. What's the top one called? No, these getting harder, huh? All right, now number 7. Number 7, let's go away from the brain. Bye-bye, brain. Bye-bye, bye-bye. On number 7, I'd like you to tell me this yellow bulge here on the side of the spinal cord. Please. That's number 7. And then number 8. I'm going to go to this picture. This is new. The tip of the spinal cord here, right underneath the ribs, the tip of the spinal cord. Number 8. And number 9. Yeah, we gotta go a little more than 10. Number 9, on this top view, what is the part of the ear called that's the round snake-like part? That's the organ of hearing. That's number 9. And then number 10, on the eyeball, what's this structure here called on the outside of the eyeball? Number 10.
Sorry, I didn't get number 8.
Number 8 was the tip of the spine here, the spinal cord. Where the spinal cord comes to a close, to a tip, there's a name for that tip.
Okay, thank you.
And then number 11, I want you to tell me on the outside where this structure is, there's a muscle here. What is that muscle called? Number 11. And then number 12. Blah, blah, blah. I'd like you to tell me, what is this pipe called here? One of these pipes. Once they split from this central one, from the windpipe. So that's number 12. That's good enough. All right. Everybody,
what was number 10 again?
Oh, number 10 is the structure on the eyeball here on the outside. This one. Good. And let's solve it. As many voices as we can get. Number 12.
Hylum.
Hylum.
Yeah, I take that. I was going for a bronchus.
Bronchus.
But I take the hilum.
Where's the hilum then?
Well, it's the whole space. It's the space. Like if you grab along and you have a place where all the vessels come in and out, and the bronchus too, and that's the hilum as a whole place. And so as I, if you put hilum for what I looked for a bronchus is totally a right answer. I could have said, you know, what's this area called, this region, and that would probably be a little more a Hylum question. And I looked at the structure that goes in and out of the Hylum, and so yeah, that might be a little ambiguous. But I would take both of them. That was a very good answer, by the way. I never had anybody tell me Hylum on a test thing like that. All right, then does that make sense? Is that okay?
Yes.
Good.
Yeah.
Then we're gonna go to the, well, first number 10 was this structure. What's that called?
Lacrimal gland.
That's the lacrimal gland. So when you see that, you know it's on the outside. And then we got the muscle right here. What would that muscle be called then?
Lateral rectus.
Good job, you guys. You're getting it. Super. That was correct. So we have lateral rectus, medial rectus, superior rectus, and the one on the bottom is the inferior rectus. And then we got the inferior oblique here where we could see it on the outside going up and down. That's a giveaway. And then on the top medial side, you got this white thing, and that's the superior rectus. I mean, superior oblique. You can only see the tendon here. Good. Just to review that real fast. And then we got the ear. What's this snail thing called?
Cochlea.
Good. The cochlea. Very good. And then there we went straight to the tip of the spinal cord. Good. Very good. Conus medullaris. That's kind of a hard word, actually, huh? That's number 8. And then number 7, I go to the cross section on the yellow thing here.
Dorsal root.
Dorsal root ganglion.
Dorsal root ganglion.
Good job. Dorsal root ganglion. And then we go to the mighty brain here. So the mighty brain here, we had those little dots. Anybody figure those out?
Superior
colliculus.
You guys are good. Yes, superior colliculus. That's excellent. What's number? What is it? Number 5. Number 5. Excellent. Oh, you're gonna all ace that test, I tell you. And then in the back, the cerebellum. Oh, sorry.
Cerebellum.
There you go. Never mind. Cerebellum. You all got that right. And then we do a little blood.
Lymphocyte.
Lymphocyte, very good. So we got monocytes, lymphocytes, neutrophils, basophils, and eosinophils. And then these are the platelets and the red blood cells. So you go with that. And then we have number 2 is this vessel right here.
Circumflex artery.
Circumflex artery, very good. Circumflex, anterior interventricular, or what is it? No, they also have a different name. I always forget. Anterior descending into circumflex. Yes. A question? No. And then last but not least, what's this one?
Left common
carotid artery.
Good, left common carotid. So you have the brachiocephalic, left common carotid, and the left subclavian. Excellent job, you guys. Well, good. Good work. With that, I think we can look at that. 7 o'clock on the dot. I think we got it. So if you guys have a question, ask now, or we're gonna get us a nice evening.
Not a serious question, but who is Schlem?
You know, I don't know, but it's just like a Yiddish word for, you know, Schlum in German is what is slum? Dirt in a gutter kind of thing. And so I think it almost sounds to me like it comes from something like that, you know, but we can figure that out. Canal of Schlem. Yeah, that would be, I know. Sometimes you wonder why these people need to put their name on these structures. Isn't that funny?
I just had a thing about the label and color. I know I'm a little bit ahead, but it's for the reproductive system. When you go to click on the electronic version to get the coloring assignment, it says that it was removed or deleted.
Oh,
so there's nothing there.
The repro?
Yes, the reproductive system.
Okay, thank you. It probably didn't do that when I updated it. All right, perfect. I will do that. Thank you.
Yep.
All right, anything else? No? Well, with that, we can go and drive home. I know, that's kind of the nice part, right? We don't have to drive home. But I'm glad you guys are in class, and I really appreciate all of you, and I wish you a nice weekend.
You as well. Thank you.
Yeah, you as well. Thank you.
Bye.
Thank you.
Thank you.
Thank you.
Bye. Have a nice weekend.
You too. Bye.